मैं राजवीर सिंह शेरान एसोसिएट प्रोफेसर इन केमिस्ट्री SRR मोरका गवर्नमेंट PG कॉलेज झुंझुनू। फर्स्ट ऑफ ऑल गुड इवनिंग टू ऑल द स्टूडेंट्स ऑफ MSc फाइनल। बायो ऑर्गेनिक केमिस्ट्री की लास्ट यूनिट एंजाइम्स वाला डिस्कस कर रहे थे अपन। इस एंजाइम्स में यूजेज ऑफ एंजाइम्स जो हैं, क्या यूज है एंजाइम्स का इंडस्ट्री के अंदर और अन्य फील्ड के अंदर, उसको अपन डिस्कस करेंगे। ये लास्ट टॉपिक है इस यूनिट का, यानी बायो ऑर्गेनिक केमिस्ट्री जो है इसका लास्ट टॉपिक है यूज ऑफ एंजाइम्स इन फूड एंड ड्रिंक इंडस्ट्री, ब्रूइंग एंड चीज मेकिंग, सिरप्स एंड स्टार्च। So, enzymes from yeast are used for alcoholic fermentation in beverage industry since they convert sugar into alcohol and carbon dioxide. Pectinases are often added to canned fruits and juice in wine industry since they hydrolyze the pectin. They make wine as well as juice clear. Glucose isomerase is used for the production of fructose and high fructose syrups from hydrolyzed maize starch, which may be used in soft drinks. Lactase is used for the prevention of lactose crystal in ice cream. Invertase is used in the manufacture of chocolates, covered berries, and other candies. Details of enzymes in food and any drink industry are discussed below. The use of Saccharomyces cerevisiaensis yeast in baking and brewing industry is because they contain enzyme for alcoholic fermentation. They metabolize hexose to pyruvate. Animal convert pyruvate to lactate under anaerobic conditions, but anaerobic ends products in yeast is carbon dioxide and ethanol. Important applications of enzyme in baking and brewing along with some other food industries are as shown in the application in the reaction. We take pyruvic acid. In presence of pyruvate enzyme, it converts into acetaldehyde, and alcohol dehydrogenases NADH and ND+. It converts into ethanol plus CO2. In bread making, first thing to be done is mixing of wheat flour with yeast and water. Starch present in wheat flour consists of D-glucose units linked by alpha 1,4 glycosidic linkage with alpha 1,6 linkage at branching points. Alpha amylase and beta amylase enzymes present in 4 cleavy. Some of the alpha 1 and 4 bonds converting starch to glucose, maltose, and some of the oligosaccharides which cannot be broken because of alpha 1,6 linkage. Glucose and maltose are metabolized by yeast, and carbon dioxide formed in the process expands the framework of dough ready for baking. As amylase is heat stable, so it may be results in too much starch breakdown and cause the bread to be somewhat soft. Therefore, wheat amylase is sometimes deactivated by brief treatment with superheated steams. Protease from asparagus oranges may be added to cause initiated breakdown of wheat proteins, which also shortens mixing time and produce smoother and more uniform dough. Sometimes malt flour is also added in dough. It also contains protease, so it may be lead to excessive gluten breakdown. Brewing for making alcoholic beverage. For brewing, starting material is malt from barley Malt is produced from barley seeds by germinating it under moist conditions. The starch present in barley is broken down by amylase to give glucose and maltose. If grains are roasted, they prevent further growth and add flavor. The insoluble material is extracted with water to produce the wort malt. So, alpha amylase from bacteria Bacillus subtilis in such more heat stable in comparison to wheat alpha amylase, which is advantage for brewing industry. It may be used to produce wort from maize and unmalted by barley, although about 20% malt is added from the outside. Now, third use is industrial preparation of glucose. In the industrial production of glucose from starch is first stabilized and partially degraded by bacterial amylase and is then subjected to fungal degradation by amyloglucosidase, which can cleave alpha 1,4 as well as alpha 1,6 linkage of starch, therefore gives food therefore gives good yield of glucose. Glucose can also be prepared by cellulosic material by treating them with enzyme cellulose from the earth. It may also be obtained along with galactose by the action of lactase. So, in jam making, invert sugar, which is a mixture of glucose and fructose, is obtained from sucrose by the action of enzyme invertase, obtained by the disruption of yeast cell wall on it. Sucrose crystallizes in presence to invert sugar at such a high concentration as required for jam making. Therefore, invert sugar is used for jam making. Now, in making invert sugar is widely used in confectionery. Therefore, it is an important substrate. In addition to action of invertase, beta fructoperoxide on cane sugar, inverts sugar may also be obtained by other enzymes. For example, the action of glucose isomerase, bacterial or fungal on glucose. Sixth use in the production of fructose syrup. First large scale production of high fructose syrup from glucose using soluble glucose isomerase enzymes was brought into light by Taksaki in 1966. Another commercial production was reported by Thomson and Collegius in 1974 by using glucose isomerase implied in DAE cellulose. Now, in clarification of cider, wines, and fruit juices, for example, apple juice and wines, etc., can be made clearly by the treatment with fungal pectinase. They cause fruit juices to clear the prevention of application of suspended particles. Pectinase are group of enzymes, for example, polyglucoronase, which break main chains of pectins and pectinesterase, which hydrolyze methyl ester. As a meat tendiger, pepin is sometimes used as meat tendiger. In South America, people wrap meat in leaves of papaya, from which pepins is obtained. Pepins and other proteases may also be used in brewing industry to prevent chill has caused by the precipitation of complex of proteins from tannin at low temperature. In cheese production, this production involves conversion of certain seasons of milk to Persian by hydrolysis catalyzed by renin, chymosin. In presence of calcium 2 plus ion, ferricin is separated from the whey after the clot is allowed to mature under controlled conditions. This solid is processed as cheese after incursions with appropriate mixture of microorganism. Now, applications of enzyme in industry. In designing fabrics, woven fabrics are sized by applying starch to the wrap threads to strengthening the yarn before weaving. When fabrics are dyed, the sizing should be removed. Now, in destaining fabrics, in dry cleaning, stains of glue, starch, or gelatin are removed by the application of certain enzymes. In dehairing hides, for the manufacture of leather hide should be made free from hair. Pancreatic enzymes may be used for the purpose which hydrolyze proteins of hair follicles. Now, in recovering silver, silver can be recovered from the photographic films by enzymes pepsin which digest gelatin. Next, in analyzing biochemicals. So, certain enzymes are used in biochemical analysis. For instance, uricase and urease are used in the determination of uric acid and urea in blood. Sucrose and raffinose contents in sugar mixture are determined polarimetrically before and after treating solutions with enzyme sucrase and maillage. In softening agent in leather industry, enzymes are also used for softening or plumping of desired skin. This process is known as wetting. In paper industry, for making the paper from wood, separation of wood fiber from each other, that is, plumping is indistinct in the subsequently spreaded into sheets. Wood fibers are bound together by lignin, which should be degraded to get chemical pulp. Pulp can also be made mechanically by tearing the wood fiber apart. From chemical pulp, lignins is removed to get brighter pulp by bleaching. Now, in manufacture of detergents, so proteolytic enzymes acylase is added to many detergents which help in removing proteinous stains from the cloth. In manufacturing of organic compounds, enzymes from bacteria are used in the industrial preparation of lactic acid, butanol, citric acid, and may other metabolisms by fermentation of carbohydrate enriched substances. Now, enzymes in synthetic organic chemistry. Enzymes can be used in organic synthesis. Two important reasons for their use in the synthetic organic chemistry are: first, they can be recycled if mounted on immobilized support. Secondly, chiral molecules can be obtained directly from achiral molecules, as shown in the following two reactions. Formation of carbon-carbon bond with the stereospecific introduction of chiral center is of special interest in synthetic organic chemistry. A large number of examples of this are available. For example, aldose catalysis aldol reactions. For this, dihydroxyacetone is first phosphorylated by kinase. In presence of aldose, ketosubstrate from ship's base with lysine residue of enzyme to give enamine intermediate which act as nucleophile and reacts with incoming chiral aldehyde. In enzyme catalyzed aldol condensation, two new chiral centers are formed specifically. High selectivity and fast rate of enzyme reactions are of great importance in the development of rapidly accessible chiral precursors. Enzymes have ability to discriminate between enantiomers of racemic substrate. Examples of enantiomeric specificity are discussed below, as shown in the following two reactions. Now, enzymes as target for drug design. Since most drugs act as modifying or blocking the activity of enzymes, a deeper understanding of suitable chosen target enzymes should lead to major advances in the rational drug designing. Following two examples will illustrate the utility of drug design as an important area of biotechnological research. For example, this TMP in bacteria and is frequently used to treat urinary tract infection, but in high concentration it attacks even human DHFR, thus becoming harmful toxic if used by the patient. In view of this, the structure of different DHFRs have been compared so that TMP with greater specificity for bacterial enzymes may be designed. Since TMP shows flexibility in its three-dimensional structure in association with enzyme DHFR, efforts have been made to synthesize TMP, which will have a rigid 3D structure in association with bacterial enzymes DHFR so that it may not be able to attack human DHFR. Now, inhibitors of renin. Inhibitors of enzymes renin are also being actively modeled. This enzyme catalyzes the first in series of reactions that leads to elevated blood pressure. Models of renin have been prepared on the basis of known structure of similar other proteins like aspartic proteinase and pepsins. This leads to the designing renin with its substrate so that native renin will not function. These inhibitors may be used for treating hypertension. In medicine, there is a strong need to develop enzymes. For examples, proteinase inhibitors, that is, the compound which have the high specificity to target proteinase and are less toxic to human beings. For example, EACA, a synthetic plasmine inhibitor process characteristics suffice to its use in medicine. Use of EACA is limited to hyperberinolysis. There is requirement to find satisfactory inhibitors for this proteinase. So this completes our unit by organic chemistry and enzymes. In the next video lecture, we will start the fourth unit, biophysical chemistry. We will start. Thank you, all of you.